So part of bacterial genetics gets to how bacteria actually regulate their genes. Because in terms of bacteria, they have evolved to be very, very efficient as far as the genes they have representing their genome. Everything in there is going to have a specific function. If that bacteria doesn't need a particular protein, it needs a way to turn it off so that it doesn't waste valuable resources making a protein it doesn't need. All right? The way bacteria do that is via gene regulation. As eukaryotes, we also regulate our genes. We have genes that are turning on and turning off all the time. Regulation of eukaryotic gene expression, way, 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 way more complicated than bacteria. All right? So that's one of the nice things about this being microbiology and not a molecular genetics class. The Lac Operon is relatively straightforward in how it works. And there are going to be some components I want you to pay attention to. And in terms of the Lac Operon, essentially what it is it's a way to control the genes that are needed to break down lactose. So you see here where it got the name, Lac Operon. What the genes associated with the Lac Operon do, it's the enzymes that are needed to break down the disaccharide lactose. So if lactose is present in the environment, then the bacteria wants to be able to use that sugar, wants to be able to break things down, needs to make the genes that are necessary for breaking down lactose. And in the case of the Lac Operon, there are 3 genes that are needed to break down lactose. All right? The names of the genes, it's LAC-Z, LAC-Y, and LAC-A. All right? One of them, the LAC-Z, actually produces the enzyme that breaks down the sugar itself, breaks down the lactose. So the gene that's produced there, it's called beta-galactosidase. All right? There is also in the Lac Operon 2 other genes. One is a permease. The permease allows essentially the bacteria to be able to pull lactose from the environment into the cellular environment, into the cytoplasm, so that it can break it down. All right? The other component is a transferase. All right? And that transferase, believe it or not, we have absolutely no idea what it is actually doing in the process of being able to break down lactose. But if we remove it, the cell isn't able to break down lactose. So it's a necessary gene. Function isn't quite known yet. That's okay. All right? I want you to know that there are 3 genes that break down lactose. All right? That's LAC-Z, LAC-Y, LAC-A. All right? And in terms of our operon, the genes are going to make up part of our operon. The other part of our operon is going to be in green here. We've got an operator region, all right? Because in terms of that operator region, this is what's going to be essentially controlling the on-off switch associated with the Lac Operon. All right? The blue box that I have drawn here, this is a promoter. All right? Remember I said when I was talking about transcription, RNA polymerase needs to know where the genes are. The promoter is that way of the RNA polymerase being able to tell where the genes are for the RNA polymerase to come in, make our transcript. Once we've made our transcript, we can make our protein. So in terms of that, what makes up the operon itself are those components, the promoter, the operator, and the genes. This is what constitutes an operon here, all right? But if you notice, I've got my pink mystery box over here. This is an important part in regulating how lactose is used, all right? This is going to be your repressor, okay? So what happens? Somewhere away from the operon, sometimes it's nearby, sometimes it's further away. Don't worry about why it's close, why it's far away. But what the repressor does, all right, that encodes a protein that can bind to DNA, all right? So the line that I have going here, this is going to represent a strand of DNA. So you're going to have A, T, C's, and G's here that make up the repressor protein, that make up the promoter region, that make up the operator region, and that make up the 3 genes, all right? So this is DNA. That repressor, let me see if I can draw this so it'll be complementary, sort of, all right, is going to have a shape something like this. And what that means, that repressor, if you notice I made some indentations here in my operator region. The repressor can bind to the operator and what that does is that inhibits transcription, okay? Because what happens, our RNA polymerase would come sit down, it's going to sit down on that promoter, it's going to start scanning looking for genes, but if it hits that repressor protein, that's going to knock the RNA polymerase off of the DNA. You aren't going to get the genes synthesized, okay? So in terms of the repressor, if the repressor is bound to the Lac Operon, that's going to have things in the off position, all right? That repressor physically interferes with RNA polymerase being able to undergo transcription to transcribe the genes. If you don't get a transcript of the genes made, you aren't going to get translation to happen. You aren't going to get the 3 proteins made. All right? So in terms of that, the Lac Operon, in the off position, you've got the repressor sitting on the operator. All right? Now, one of the key things with the Lac Operon it's considered an inducible system, all right? And in order to induce transcription, we've got to have lactose present, all right? My next video, I'm going to show you what happens in order to turn that Lac Operon on, okay? In order to turn the Lac Operon on, we're going to need to have lactose present. So this video is just to kind of give you an introduction to the portions of the Lac Operon. I want you to know all of those components. The repressor I drew up here because it was easiest to kind of start with off. And then my next video, I'm going to show you how that repressor now is going to be removed from the operator region so that we can turn on transcription of the genes. All right? If you have any questions, please